Our project implements a comprehensive bioinformatics pipeline for analyzing the pangenome of baker's yeast, which is Saccharomyces cerevisiae. For those unfamiliar with the concept, a pangenome represents the entire genetic information of a species, encompassing all genes found across different strains. We are integrating Syntheny block analysis with functional annotation to identify conserved genomic regions and their associated biological functions. Synteny blocks are regions where gene order is preserved across different genomes, suggesting evolutionary constraints on genome organization. This approach allows us to reveal strain-specific genetic elements and their potential roles in phenotypic diversity among yeast strains. By understanding which genes are conserved across all strains versus those that are strain-specific, we can gain some insights into core cellular functions versus adaptive traits. Our pipeline includes several key components, which are the genome preprocessing, gene prediction, Synteny analysis, and functional annotation using InterProScan. Through this work, we are gaining insights into the evolutionary relationships and functional diversity within the Saccharomyces cerevisiae species, which has implications for both data science and for both basic science and biotechnological applications. Here's an overview of what I'll cover today. I'll start with an introduction to the concept of pangenomes and synteny analysis, followed by our specific project objectives. I'll then provide some background information on Saccharomyces cerevisiae as a model organism, and then I'll discuss our methods in detail. Then I'll present our current status and finish with a summary of our findings and future directions. The Saccharomyces cerevisiae, which is also known as baker's yeast, is a model organism in genetics and molecular biology. The concept of a pangenome represents the entire gene set of all strains within a species. It's a relatively new paradigm in genomics that moves beyond the limitations of a single difference genomes. The pangenome is typically divided into three components. First, the core genome, which consists of genes present in all strains of the species. These genes typically encode essential functions required for basic cellular processes. Second, the accessory genome, comprising genes present in some but not all strains. These genes often contribute to the strain-specific adaptations to particular environments or lifestyles. And third, strain-specific genes, which are unique to individual strains and may confer highly specialized traits. Pangenome analysis helps us understand genetic diversity and adaptation mechanisms within a species. It provides insights into how organisms evolve in response to different selective pressures and how genetic variation contributes to phenotypic diversity. Synteny blocks are conserved regions with preserved gene order across multiple genomes. By identifying these blocks, we can gain some insights into genome evolution, structural rearrangement, and functional constraints on genome organization. The maintenance of a gene order over evolutionary time suggests that there may be some functional advantages to keeping certain genes together, such as co-regulation or physical interactions within a gene products. Our project combines synteny analysis with functional annotation to gain deeper insights into yeast genome evolution. This integration allows us to not only just identify the conserved regions, but also to understand their functional significance. By correlating synteny conservation with the functional annotations, we can test a hypothesis about the relationship between genome structure and function. Now here's an introduction to Saccharomyces cerevisiae, which is commonly known as baker's yeast. It is one of the most well-studied model organisms in genetic and molecular biology. It was the first eukaryotic organism to have its genome completely sequenced back in 1996, which is a milestone achievement that revolutionized genomics and paved the way for numerous advances in our understanding of eukaryotic cell biology. Despite its relatively small genome size of approximately 12 million base pairs, Saccharomyces cerevisiae contains about 6,000 genes, many of which have human homologs. This makes it an excellent model for studying fundamental cellular processes relevant to human biology and disease. Our project has several key objectives that guide our approach and implementation. First, we aim to develop a comprehensive pipeline for pangenome analysis of Saccharomyces cerevisiae strains. This pipeline needs to be modular, efficient, and reproducible. Modularity ensures that different components can be updated or replaced as methods improve, while reproducibility is essential for scientific validity and for allowing others to build upon our work. Second, we want to identify and characterize synteny blocks across multiple yeast strains. These blocks represent evolutionarily conserved regions that may have functional importance. We are interested in understanding the distribution of these blocks across the genome, their size distribution, and how they relate to known functional elements like genes and regulatory regions. Third, we are analyzing the functional annotations of genes within and outside synteny blocks to understand the relationship between conservation and function. Do synteny blocks contain genes with particular functions? Are certain functional categories more likely to be found in conserved regions? These are the questions that we aim to address. Fourth, we are determining the core and accessory genome components of Saccharomyces cerevisiae to understand the genetic basis of strain diversity. This involves identifying which genes are present in all strains versus those that are strain specific or present in only a subset of strains. We are also interested in the functional characteristics of these different components. Fifth, we are investigating the relationship between genomic conservation and functional importance. Are highly conserved regions also functionally critical? This question gets at the heart of the relationship between the evolutionary conservation and biological function, a fundamental question in evolutionary biology. Sixth, we are creating visualizations to represent synteny patterns and functional distributions, making our findings more accessible and interpretable. Effective visualization is crucial for understanding complex genomic data and for communicating our findings to others. And finally, we are stabilizing a reproducible workflow for future comparative genome studies which could be applied to other organisms just beyond yeast. While our current focus is on Saccharomyces cerevisiae, here comes the background of our project. So it's mainly divided into three categories. First stage is pan-genome concept, second stage is about synteny analysis, and third stage is about discussing Saccharomyces cerevisiae as a model. Synteny analysis focuses on the conservation of gene order across genomes. Synteny blocks are regions with preserved gene content and order, which can reveal evolutionary relationships genomic rearrangements, and functional constraints on genome organization. The conservation of gene order over evolutionary time is not a given. Random genomic rearrangements would be expected to shuffle gene order over time, unless there are selective pressures maintaining a synteny. Such pressures might include functional constraints like oppedons through these are layered in eukaryotes, co-regulation of genes, or physical interactions between gene products. Breaks in synteny can indicate sites of genomic rearrangements like inversions, translocations, or insertions of mobile genetic elements. These rearrangements can have functional consequences and potentially disrupting gene regulation or creating new gene combinations that affect a phenotype. Saccharomyces cerevisiae is an ideal model for this type of analysis for several reasons. It has a compact genome with relatively few introns, making gene prediction more straightforward compared to many other eukaryotes. There are diverse strains available with different origins, including laboratory strains like S288C, which is our different strain, clinical isolates like YGM789, vine bear strains like RM111A, and wild isolates like Y55, BJ4, and HN1. These diverse strains have adapted to different environments and lifestyles, in leading to genetic divergence. Laboratory strains have been selected for traits that make them amenable to laboratory manipulation, while wild strains retain adaptations to their natural habitats. Industrial strains used in brewing and baking have been selected for traits beneficial for these processes. This diversity makes Saccharomyces cerevisiae an excellent model for studying the genetic basis of adaptation and the relationship between genotype and phenotype. Now let me walk you through our methodology in detail, explaining the key components of our pipeline and the tools and approaches we have implemented. For data collection, we selected six diverse Saccharomyces cerevisiae strains to represent different ecological niches and evolutionary histories. S288C serves as our reference laboratory strain and is the most well-characterized yeast strain with extensive functional annotation available. YGM789 is a clinical isolate providing insights into adaptations that might contribute to pathogenicity. RM111A is a wine strain that has evolved in the context of wine fermentation. BJ4, Y55, and HN1 represent diverse ecological origins, adding breadth to our analysis. We obtained these genome sequence from the NCBI genome database, ensuring that we had both genomic sequences and annotation files for each strain. Our pre-processing steps were crucial for ensuring consistent and accurate downstream analysis. We began with genome sequence standardization to ensure uniform formatting across all the strains available. This involved converting all sequences to the same format. Following standardization, we performed a gene prediction to identify coding regions in each genome. For this, we used Augustus, which is a gene prediction tool specifically trained for fungi, including Saccharomyces cerevisiae. Augustus employs a hidden Markov model approach that incorporates species-specific parameters with codon usage, splice site patterns, and intron length distributions. The output from Augustus includes both gene structures and predicted protein sequences. We carefully validated these predictions by comparing them to known annotations for the reference strain and by checking for features like proper start and stop codons, realistic gene lengths, and the absence of premature stop codons. After gene prediction, we parsed and standardized the GFF annotation files which contain information about gene locations and features. We developed custom parsers to extract relevant information from these files and to ensure consistent formatting across all strains. We also translated the predicted genes to protein sequences for the functional annotation. For synteny analysis, we performed a BLAST-based all versus all comparison of genomes. BLAST, which is also known as Basic Local Alignment Search Tool, allows us to identify regions of similarity between sequences, which is the foundation for detecting syntenic regions. We set specified parameters including an e-value threshold of 1e-10, which controls the stringency of the matches, a minimum alignment length of 100 base pairs to show that we can ensure substantial similarity, and a minimum identity of 75% to focus on well-conserved regions. Our synteny block identification criteria were designed to balance sensitivity and specificity. We allowed a maximum gap of 5 genes, recognizing that small insertions or deletions might occur within otherwise syntenic regions. We required a minimum of 3 genes in conserved order to define a synteny block, as shorter conserved regions might arise by chance. For pangenome analysis, we used ROADY, which is a high-speed bacterial pangenome pipeline. Although ROADY was designed for bacterial genomes, we adapted it for our yeast analysis by carefully formatting our input files to match ROADY's requirements. This involved converting our gene predictions and annotations to a format that ROADY could process and adjust. ROADY works by clustering genes based on sequence similarity and determining their presence or absence across all the strains. This shows that which genes are present in which strains, allowing us to identify core genes, accessory genes, and strain-specific genes. For functional annotation, we extracted protein sequences from our gene predictions and submitted them to the InterProScan web service. InterProScan is a powerful tool that integrates various protein signature databases to provide comprehensive functional annotations. It searches against databases like PFAM, ProCite, and Gene Ontology, providing a rich set of functional annotations for each protein. The InterProScan analysis gave us a gene ontology, which are GO terms which describe gene functions in a standardized vocabulary across 3 domains: molecular function, biological process, and cellular component. It also provided information on protein domains and families, enzyme classifications, and pathway information. We then performed statistical analysis of function enrichment to identify overrepresented functions in different parts of the pangenome. This involves comparing the frequency of a specific geoterms of a protein or protein domains in the core genome versus the accessory genome, and identifying statistically significant differences. We used a standard statistical test like Fisher's exact test with multiple testing correction to ensure the robustness of our results. Finally, we integrated our synteny analysis with the functional data to investigate the relationship between conservation and function. This involves some mapping functional annotations onto synteny blocks and analyzing whether certain functions are enriched in conserved regions or not. We implemented all of these analyses in Python using libraries like PyOPython for sequence handling, pandas, and for data manipulation, matplotlib, and seaborn for visualization. Our implementation emphasizes modularity, allowing different components to be updated or replaced as methods we can improve and reproducibility, ensuring that our results can be validated and built upon by others. Let me now share our current status, highlighting that what we have achieved so far and the insights we have gained from our analysis. We have successfully established a GitHub repository with a clean, well-structured code. This repository serves as both a record of our work and a resource for others interested in pangenome analysis. We have organized our code into logical modules corresponding to different steps in the analysis pipeline with clear documentation and examples to facilitate use by others. Our results so far include the identification of approximately 4,500 core genes present in all 6 strains we analyzed. These core genes represent about 75% of the total gene content of each strain, indicating a substantial shared genetic foundation across diverse Saccharomyces cerevisiae strains. The remaining 25% comprises the accessory genome with about 1,500 genes present in some but not all strains, and a smaller number of strain-specific genes unique to individual strains. We have also detected around 200 conserved synteny blocks across the 6 strains, which show interesting patterns of conservation. These blocks range in size from our minimum threshold of 5 kilobases up to several hundred kilobases, with an average size of approximately 50 kilobases. They are distributed across all 16 chromosomes of Saccharomyces cerevisiae, but not uniformly. We have also analyzed geoterms and protein domain distributions, finding that core genes are enriched for essential cellular functions. Specifically, we conserved a significant enrichment of geoterms related to translation, transcription, DNA replication, cell cycle control, and central metabolic pathways in the core genome. This aligns with a revolutionary theory which predicts that genes involved in fundamental cellular processes should be conserved across all strains of a species. In contrast, accessory genes show enrichment for functions related to stress response, secondary metabolism, transport, and cell wall organization. These functions are likely involved in adaptation to specific environments, explaining why they are not universally conserved. For example, genes involved in galactose utilization are present in some strains but absent in others, reflecting different carbon source availability in different habitats. We have also established correlations between the synteny conservation and functional importance. Our analysis shows that genes within a conserved synteny blocks tend to have more fundamental cellular functions compared to genes outside these blocks. They are more likely to be involved in essential processes and to have more protein-protein interactions. This suggests that the maintenance of a gene order in these regions may be functionally significant, possibly due to co-regulation or the physical interactions between gene products. Our project documentation is comprehensive, including a detailed README with data collection and instructions and code organization optimized for readability and maintenance. We have also made sure to document not just the code itself, but also the underlying biological concepts and analytical approaches, making our work accessible to researchers with varying levels of computational expertise. To summarize our work, we have developed a comprehensive pipeline for Saccharomyces cerevisiae pangenome analysis that successfully integrates synteny block detection with functional annotation. This integration provides a more complete picture of genome evolution and function than either approach alone. We have identified code and accessory genome components across 6 diverse strains, revealing the genetic basis of shared and strain-specific traits. The core genomes, comprising about 4,500 genes, provides the fundamental cellular machinery shared by all Saccharomyces cerevisiae strains, while the accessory genome contributes to the unique adaptations of each strain. We have also detected conserved synteny blocks and their associated functions, providing insights into the relationship between genome organization and function. The conservation of gene order in these blocks suggests functional constraints that have maintained a synteny over evolutionary time. Our approach has revealed interesting patterns of genome conservation and functional distribution that contribute to our understanding of yeast evolution and adaptation. We have shown that core genes tend to be involved in essential cellular processes, while accessory genes often contribute to environmental adaptation. We have also demonstrated that genes within synteny blocks are more likely to have fundamental cellular functions. The visualizations we have created effectively represent these genomic conservation patterns and make our findings accessible to others. These include heat maps showing synteny conservation across lanes, bar charts depicting functional distributions in different genome components, and network diagrams illustrating relationships between synteny and function. We have established a reproducible workflow for comparative genomics that can be applied to other organisms in the future. While our current focus is on Saccharomyces cerevisiae, the methods and approaches we have developed could be adapted for studying the pangenomes of other species. Here are the key references that informed our work. These include foundational papers on the pangenome concept, the original Saccharomyces cerevisiae, a genome sequencing paper, studies on the yeast population genomics. Thank you.